Hi guys, welcome to my YouTube channel Unique Bio Classes based on NCERT syllabus for PU first and second year. If you like my channel, please subscribe, like and share and hit the bell icon so that you can get notifications as soon as videos are getting uploaded. Okay guys, if you like my channel, please subscribe and don't forget. Fine, today we shall start a new session on inheritance of one gene. In previous class we have started with the principles of inheritance and variation. So this inheritance of one gene you will find on page number 71 in your NCERT textbook. So this inheritance of one gene, it was studied by, or it was given by Gregor Johann Mendel. Okay, what he did was, first we shall go through, right? What he did, he chose plants. Okay, which plant? As you know, we know that it is a pea plant. Okay, Mendel chose a pea plant. Now in that pea plant, I shall give this as one plant and this one as another pea plant. Okay, one is female, another one is male. When he chose the pea plant, in that he chose one as a tall plant, another one was dwarf. Okay, tall and dwarf. So which character we have chosen? It is nothing but stem height. Okay, when we are talking about tall and dwarf, it is a character stem height. These are the two contrasting characters of that stem height. Okay, so when he has chosen about these two plants, he has cross-pollinated it. Cross-pollinated in the sense, now we have taken this plant as a female plant, which means we want a gynoecium of this plant, of this flower, and androecium, that is stamen, which carries pollen grain, that one from this flower. Okay, so what we studied in previous chapters, in the second chapter, like artificial hybridization, like emasculation and bagging, we are collecting the pollen grains from here and dusting over it on the stigma of this flower. Okay, you can choose any of the flower as a female or a male, not restricted that a tall plant should be female or a dwarf plant must be a male. It is not like that. Whichever you want, you can choose. Fine. Now, when we have collected pollen grains from this flower and dusted it on the stigma of this another flower, then what happens? Then it can undergo fertilization. After fertilization, what does it will do? It will produce seeds. Okay, fertilization will, after fertilization, zygote formation and formation of an embryo. So all that process will take place. It produces a seed. Okay, so now when here it has been deposited, after that what seeds has been produced, the F1 generation, what he found, the plant which has been produced in his field, he saw that plant was tall. Okay, in his field what plant was grown, he saw that it was tall. Now this tall plant was self-crossed. Okay, self-crossed means the same plant. This is a bisexual flower. It carries both androecium and a gynoecium. So then from the same androecium or a stamen, pollen grains are collected and it was dusted it on the stigma of the same flower. Then what it did after self-pollination? Okay, then after self-pollination, what happens? Again fertilization takes place, it produces the seeds. Those seeds were sown under the soil. Okay, when these have been sown under the soil, he found that there was growth of tall plants and dwarf plants. Okay, now these are the parents. These two are the parents. Which one? The first we have choose tall and a dwarf plant. These are the parents. Then he got F1 generation. For this we call it as F1 generation. F is what? Filial generation. First filial generation is this tall plant. When he has done all this, sorry, next one we have got F2 generation. Second filial generation. Okay, this was second filial generation. Now, when he has crossed this, he found that in the F1 generation, there was a similarity in the tall plant, which means the F1 generation was having same like of a tall plant, not of a dwarf plant. Okay, as many, I am showing here only one plant. It doesn't mean that only one seed will be produced by these plants. It has produced many of the seeds, and in those seeds, all the seeds, when he has sown under the soil, then he found that all those plants were tall. Okay, which means in F1 generation we are getting only tall plant, not the dwarf plant. So for his surprise, he was surprised to know what might be the reason behind this to get only tall plant. Where does the dwarf character has been gone? According to our view, when you see this tall crossed with a dwarf, dwarf means, as you know, it is nothing but short in common terms. When tall has been crossed with a dwarf, we have to get medium. Is it? we think of medium. Okay, medium sized plant. But what he got only tall plant. So he was amazed to see why only tall plant, what has been happened to a dwarf plant, or what has been gone through because of which he has got only tall plant. So for that reason he has self-pollinated the same plant, and he found that in the F2 generation both the characters of parental type were visible in the F2 generation. That few plants were tall, few plants were dwarf. Okay, so to analyze this, here we have this one here in the diagram. Okay, so this is the pea plant flower. So this pea flower, it do have petals, there is a stigma, anther, stamen and a carpel. It is very clearly visible. Now here you can find this is a one of the parent female, and this is another parent male. So in the female parent, with the help of a scissors, anthers are removed. Okay, when removal of anthers with the help of scissors, what we call it as emasculation. When it has been removed, this is a female flower. We don't want its anther, we want its gynoecium. That's why it has been removed. Anthers have been removed, which means this step is emasculation. Now this one is a male flower from which we are collecting the pollen grains. That pollen grains have been transferred or dusted it on the stigma. Now you can find that this both the plants after fertilization will produce seeds. Now these seeds, when it has been sown under the soil, they will germinate and they'll produce the plants. And those plants, for his surprise, he found all the plants were tall. Okay, when he self-pollinated, when he got these plants, after this what he did, he self-pollinated these plants. Then in F2 generation he got both tall and a dwarf. These are steps in making a cross in pea. Okay, this was just a general idea what he has got. And not only he has used this stem height character tall and a dwarf, he has used some other characters also. He found the same result as we are going to get in the stem height in a tall and a dwarf. Now same one he has shown here. Just one problem is that I have a habit to write a lot of things in the textbook, especially in NCERT. The space which has been provided here, this space will be used to write the information which you wanted to add some more information. So I will be using that area to write. So if you are getting confused with that and what has been printed, please refer your textbook, and while referring your textbooks you can see this, so that it will be very easy for you. Anyhow, in the next page you can find it has been given figure 5.3. It is a diagrammatic representation of a monohybrid cross. In this monohybrid cross, what you will find, there is a parental type. Okay, so parents, what parents we have used? Tall, and one was dwarf. Clearly it is visible. One plant is tall, another one is dwarf. Again, what we are doing, same step what previously it has been shown, emasculation, okay, and dusting of pollen grains. Both it has been done. So after that what you will get, F1 generation. In F1 generation, all the plants we have got tall. Then all the in that it has been self-crossed. Either you can self-cross like this within two plants, but it is of the same plant. Then you can find after selfing, we found that all the plants in F2 generation, 3 plants are tall, 1 is dwarf. Okay, which means 75% of the plants were tall and 25% of the plant were dwarf, which means here you can find that the parental character which was not visible, dwarf character which was not visible in the F1 generation, it has been visible in the F2 generation. Okay, you can find clearly it is present in the F2 generation, not in F1 generation. Now when he found this, what might be the reason behind that only tall is expressing in F1 generation, why not dwarf is expressing in the F1 generation? That was his question. And why that dwarf is expressing only in the F2 generation, why not in F1 generation? Okay, why it is visible in F2 generation? That was his question. Okay, for all this he has done many of the observations by using many of the plants, and he found that there was something was getting stably passed on. And stably passed on means something which is present in this plant, it is without getting disturbed, without getting any changes, it is getting transmitted or transferred from parents to offspring. Okay, what was that? He called it as factors. Okay, what it was getting transmitted, what was getting transferred from parents to offsprings, he called it as factors. Okay, these factors are those which are present in the parents and which will be transferred to the offsprings without getting any changes in those. That has been given by Mendel. But these factors we know today as genes. Okay, these factors we know today as genes. These genes are getting inherited. These genes are getting transferred from parents to offsprings. But the term gene was not given by the Mendel. He has used the term called as factors in place of genes. Okay, hope you understood this. Fine. Now in this gene it consists of what? A pair of alleles, and this pair of alleles are sometimes it might be similar or sometimes it might be different, as we have given about homozygous and heterozygous. Okay, this what I have discussed till now, it was just an a theoretical explanation of it, how he did all this. You have to imagine all this he is doing in his field. Okay, it takes lot of time. It doesn't mean that only we are doing it on the paper and showing it so easily. It's not that easy. He is doing all this in his field. He chose one plant which is tall, he chose one plant which is dwarf. Okay, he has selected it, then he has done with a emasculation and a bagging technique. After that, when it has produced a pollen grain, that pollen grain has been dusted it on the other plant, then it has to germinate, then it has to produce seed. We have to wait, is it? Then it will produce those seeds. Those seeds it has to undergo a drying phase. Okay, then after getting dried, those seeds will start to germinate in a favorable condition, especially in rainy season. Then when it is sown under the soil, then it will germinate. Then we are going to get these plants, and if we are getting flower, then you are going to see the height of the plant. Okay, which means it takes lot of time. Hope you got this. Okay, then continuously again when it has been self-pollinated, same story will get repeat again. It has to get fertilized again, it has to produce seeds. Seeds should undergo a drying process, then it has to get a favorable conditions to get germinated. Again you will get F2 generation. Similarly he got F3 generation and F4 generation also, but it has been restricted only to F2 generation because same results he have got for F3 and F4. Okay, so this was a theoretical explanation of it. Now I shall go to the how he has crossed it, how he has used it. We shall see here. Now here we were talking about this inheritance of one gene. By using these plants, by seeing these plants, we can use alphabets for each gene. We can use alphabets for each gene. What alphabets we are using? First, okay, I will show you here the alphabets which has been used. For tall, capital T is used. For dwarf, small t is used. Okay, these are the alphabets. Now capital T, capital letter has been used for those which has been that character which will be expressed in the F1 generation, and small t is expressed for the other character which is not expressed in the F1 generation. Hope you got this. Now in F1 generation we got tall, so that will be capital, whereas the other which didn't expressed in the F1 generation, we are giving it with the small. Now the alphabets which have been used here, we are writing it twice. Capital T, small t, small t. Why we are writing it twice? Why not once? Because we are diploid. First one is the organisms are diploid and they do have a presence of alleles. Okay, a pair of alleles with okay, we have a pair of alleles. So when we having a pair of alleles, so to represent that we are giving that one in a pair. Okay, that is capital T, small t, small t. In place of capital T or a small t, you can use some other alphabets also, but it creates a confusion among the students, so that's why we don't suggest to use some other letters. So it's better to use these alphabets only for further crossing, which I'm going to give it here. Okay, so now I shall give how to cross this. Right now I shall give first here as parents. Okay, parents, what parents we have chosen? That is, one is female, another one is male. If you start writing all this simultaneously, then it will be very easy for your understanding. Okay, then we are going to write parents phenotype. Hope you remember phenotype. Okay, in the last session we have discussed a physically expressed external character in an individual. Okay, in an individual, the physical appearance when it has been given, for that we call it as phenotype. Now what is the phenotype? One phenotype we have chosen it as tall, another phenotype is dwarf. Okay, so what character we have chosen? The character which has been chosen is stem height. Stem height is the character. Okay, what are its two contrasting characters? It is tall and a dwarf. These are the phenotypes. Phenotype of the parents. Now, genotype. Now, what is this genotype? Genotype, in previous session we have discussed genetic makeup of an individual for a phenotype. Okay, genetic makeup. What will be the genes of this phenotype? What we used? Letters. We used capital T, capital T. For dwarf, we have used small t, small t. Right? So after this, what we are going to do now, this is the phenotype and genotype, and then after this we have to cross this by using artificial hybridization. Again, what we have seen in the flats in the diagrammatic representation, same you can find here. Then what this plant will do, these plants will produce gametes. Now female plant will produce what? It will produce egg, is it? Now it will produce an egg which carries one of it, because gametes are haploid, not diploid. Okay, so here I will give here as gametes. Okay, these are gametes. It is capital T and this one is small t. What is the gamete of the male? It is pollen grain. Pollen grain will carry the male gamete. Okay, egg is the female gamete which will be present in the embryo sac. Now I have shown only one. You can, if you want, you can show two different eggs, two different pollen grains, because only one pollen grain will produce? No, definitely not. Many number of pollen grains will be produced, as many number of pollen grains will be produced in a homozygous condition that will be having similar gene in a gamete. So similar gene in a gamete here you also you can find. Now rather than creating confusion, that's why because both are similar, we are using only one for further cross. Okay, after formation of gametes, what we'll find? There will be a fertilization. I will take only one egg and one pollen grain. This we call it as fertilization. After fertilization, what will happen? Zygote is formed. Now that zygote, what it will carry? It will carry, zygote is diploid. So one gene from here and one gene from here. That is capital T, small t. For this, what we call it as first filial generation or F1 generation. First filial generation, or if you write only as F1 generation or F1 generation also, it's okay. You can write this also. So this is F1 generation. Okay, now this is the genotype of the F1 generation. What will be the phenotype? What you have observed in the field when we have crossed tall and a dwarf plant? What was the F1 generation? Either the plant was tall or a dwarf. It was tall. Okay, it was tall. Then after self-crossing, okay, selfing or self-pollination. Self-pollination, how we did, as you have seen earlier, that pollen grain from one plant to the other plant of in a same flower, that it has been crossed. So when we self-cross, again this will become a parent now. Now it will produce two different types of gametes, capital T, small t. Here we have shown only one type of a gamete because it was homozygous. For this we call it as heterozygous. This is homozygous and heterozygous. Definition of those I shall give it later. So now homozygous will produce one type of gamete, whereas heterozygous will produce two different types of gametes. If I am talking about self-crossing, if I am choosing one more here, self-crossing, I am taking capital T, small t, one more plant or from a same plant, then you can find, you'll get capital T, one gamete, small t, another gamete. Okay, these are the two gametes which you will get it here. Okay, now after production of gamete, what happens? There is a fertilization. Now what you have to do, you have to be careful, and it is very easy if you understand once. Now what parent, now this is a female parent, this is a male parent. If you just imagine like that, female parent egg is not fusing with the egg, right? Egg will fuse with the pollen grain. Okay, male gamete. Female gamete will fuse with a male gamete, or a male gamete will fuse with a female gamete. You no need to cross something like this. Okay, female gamete is not fusing with a female gamete. Do remember this point. So what happens? You are crossing like this. So what you will get here? You will get a zygote of capital T, capital T. Okay, then if you take same capital T, then you are crossing with this. What you will get? Capital T from here and small t from here. Did you got? Again, this is small t, is crossed with capital T. Always we'll write capital first, right? So that's why capital T, small t, then here small t, small t. Okay, so now when we have seen that F1 generation, it was tall here, because why we are giving tall, just now we have seen, we have studied theoretical part of it. We said that F1 generation, what Mendel has observed in his field, that all the plants were tall. Based on that we gave tall here. Okay, not only based on that, here when we say it as tall, the other one will be dominant and other will be recessive. Hope you remember dominant and recessive. A gene or a character expressed in an homozygous condition of an organism. Okay, now which character is expressing, which gene is expressing, capital T is expressing. What it is expressing? It is expressing it as tall. This small t is not expressing. Both capital T and small t is there, but small t is not expressing, only capital T is expressing. That's why plant has been produced as a tall. Whereas here now you can find the capital T means it is very clear it is tall. Small t means it is very clear it is dwarf. Now capital T small t means what? It is tall, and this is also tall, which means 3 plants are tall. These 3 plants are tall and 1 plant is dwarf. So what ratio you will get? 3:1 ratio. Okay, 3 plants are tall, 1 is dwarf. Now how we are giving this as tall? As I said, based on dominance and recessive character, the one which express, okay, from both the characters, contrasting characters of a stem height, the contrasting character which is expressed in the F1 generation, for that we call it as dominant. Okay, because another one is not expressing it. One which is expressed dominant, another which is not expressing it is recessive. So similarly you can find that here when both are capital, then it will express it is tall. When both are small, it is recessive, it is expressing as dwarf. If one capital and one small, if it is there, which one will express? The capital letter will express that capital is dominant. So we are using capital for what? Tall character. So it becomes tall. Okay, so this is how you are going to write. Now here you are going to give F2 generation. Okay, this one will be F2 generation. Now for this, what you will write? Gametes of F1 generation. For this you will write it as gametes of F1 generation, and for the next one you will write F2 generation. Okay, after crossing this, what you are going to write? You are going to write phenotypic ratio. Okay, we are giving phenotypic ratio and genotypic ratio. Okay, phenotypic ratio and genotypic, these two ratios are very important, especially for inheritance of one gene. Phenotypic ratio is what? Phenotype, physical appearance. For while seeing 3 are tall and 1 is dwarf, right? So what you will write? 3:1. 3 are tall, 1 is dwarf. Okay, when we write genotypic ratio, we are saying genotype. Capital T, capital T. How many capital T you have here in the F2 generation? We have only 1. Okay, then we have 1 capital T, capital T. It is homozygous tall. Homozygous tall. 1 we have homozygous tall, and 1 we have homozygous what? Homozygous dwarf. So what is there left in between? Okay, capital T, small t. We have capital T, small t. Those are 2. So for that we write it as heterozygous. Heterozygous tall. Okay, so recessive condition, never it will express in heterozygous. Recessive will express only in homozygous. Dominant will express in homozygous and heterozygous. Okay, this ratio, genotypic ratio, is 1:2:1. How you are saying? You are seeing the genotypes of the individual. One is having capital T, capital T. One is having small t, small t. 2 are having capital T and small t. Now homozygous means individual having similar genes. Okay, individual having similar genes in an allelic pair. Heterozygous means individuals having different genes in an allelic pair. Okay, these are 2 different genes. which is producing two different types of gamete. These are two similar genes which will produce same type of gamete. Okay, hope you got this. Now this is how you are going to cross. You will write about this. Okay, it is a inheritance of one gene or monohybrid cross. Okay, why we say it as monohybrid cross? Because inheritance of one gene. Why we are saying it as one gene is getting transmitted. We are talking about one gene, which is that gene, stem height gene. Stem height gene is having what? Two contrasting characters. Okay, one is tall and another one is dwarf. Because one gene is getting inherited, for that we call it as inheritance of one gene. Even that is called as monohybrid cross. Okay, why we call it as monohybrid cross? Mono means single, hybrid cross. Okay, single hybrid cross. We are using only single character, one character which is getting transmitted. Okay, oh fine. Now in this you will find what F2 generation I have shown. F2 generation, the way I have shown in this way, the way I have used by crossing it, you can use either this method or even you can use the checkerboard. You can use the checkerboard as you can find it here in your textbook on page number 73. You will find it here. See, they have chosen a tall and a dwarf plant. What gametes I have shown here, capital T. Okay, these it has been shown here, and small t it has been shown here, and F1 generation in the bracket. Okay, in the square box. Okay, for this box we call it as Punnett square. This Punnett square was given by a British genetist called as Reginald C. Punnett. By his name, this Punnett square has been, the name Punnett square has been given for it. Now here I have shown only one F1 generation. Here they have shown 4. Why? Because they have crossed all the all the gametes, but I have chosen only one to show one plant because all are similar. That's why I have used only one. If you use only one, no deduction of marks takes place, so you be free of that. If you want, you can write. If not, you can leave it. Then in F2 generation, again it has been self-crossed. Again similar square, Punnett square has been given with the same capital T, one small t, one two heterozygous conditions. Okay, now how to plot that I shall show you. Again it will get confused here how to write it. I will show you here once again. Okay, I shall use some other fine. Now in this I will show you all that F1 generation. What was the F1 generation? We have got capital T, small t. Okay, we have shown its gametes as one as capital T, another one as small t. It was self-crossed. We have got with another plant, so we have got capital T, small t. These 4 gametes we have got in F1 generation. Now we have crossed like this. Okay, in the previous one, what you can do is you can plot a box, something like this. Okay, now on the one side we are showing female and a male. Okay, we'll show like this, female. Okay, column for one parent, another row for one parent. Okay, one row for one parent, one column for another parent. Here one we shall use. Now here it has been given male. I will use these two because both are similar. One capital T, small t, and here from other parent capital T, small t. We are writing gametes here. Okay, this method you are using in your mathematics, I think. So if you are good in mathematics, then you will come to know about this also. Here capital T from here and capital T here. What will get capital T from here and small t from here. Again capital T, small t, small t, small t. Okay, so this is how you are going to write. This one is nothing but F2 generation. How you are going to plot it. Okay, you are giving the square in that one row and one column for one gamete's parent, one parent's gamete, and another row for another parent. Okay, this is how you are going to do. Rather than crossing like this, if you are getting confused in this, then you can choose this method also. If you do this, that is also correct. If you do this, that is also correct, but choose whichever you feel better. Okay, which one is easier. And here itself you can write this is tall, this one is also tall, this is tall, and this one is dwarf. Then phenotypic and genotypic ratio. And with this, you have one more about that Punnett square. You have in your textbook on page number 73. Here in your textbook you will find Reginald C. Punnett. It was developed by a British genetist. He was a genetist. It has been asked in the entrance exam whether he was a genetist or some other. It has been asked. So please do remember genetist Reginald C. Punnett, name of the scientist. What is this Punnett square? It is a graphical representation to calculate the probability of all possible genotypes of offsprings in a genetic cross. This will be a definition of it. Okay, we are representing in a graphical form so that we can find all possibilities of offsprings easily. Okay, so this will be the definition of that which you find in your textbook. Okay, then you can find in your textbook about law of dominance and law of segregation. I have prepared a note of it. Let me show you. Okay, fine. Now, this is inheritance of one gene or monohybrid cross, what we have crossed just now. Its definition is hybrids produced from parent differ in a pair of contrasting characters. Differ in a, which means differ in one pair of contrasting characters. Okay, hybrids has been produced from one parent which are differing in pair of contrasting character. For that we call it as inheritance of one gene or monohybrid cross. Okay, so whenever they ask, we have got this question, explain inheritance of one gene. What you will do, you will write this definition, then you will cross this, okay, till where you will write till phenotypic and genotypic ratio completely, writing this homozygous, heterozygous, everything till here you are writing this. Okay, this is the schematic representation of that. If they ask with the explanation, you have to write and explain it. Okay, and this will be asked for 5 marks. Very important question these are. Okay, and with the same question, with the same graphical, what the schematic representation we have shown just now, with the same, they are going to ask with another question, that is law of dominance. Here we have law of dominance is what? When two contrasting characters are present in a single individual. Two contrasting characters, what we used? Tall and a dwarf. In a single individual, one character is considered as dominant and other is considered as recessive. Is it? One is considered as dominant, one is expressed, and other one is as recessive, as unexpressed, as we found here. Okay, we choose two contrasting characters. Is it? We choose one character with the two contrasting characters, and in that one was dominant which was expressed and other was recessive which was unexpressed. To explain that, this is law of dominance. Okay, one where the dominant will express and the recessive will won't express. That law of dominance also was given by Gregor Johann Mendel by using the same example. Definition is here, example is here, the schematic representation. Just explanation will get different. While explaining, you'll stress more on the dominance character, which one is expressing, which one will won't express, why it will express, why it will won't express, that you will explain while explaining about the law of dominance. Now one more law we have, second law. Okay, this law of dominance is called as first law. This law of segregation is the second law. These are the two principles of inheritance. Okay, in that. Now first one and second one is law of segregation. When a pair of character is brought together, right, when a pair of character is brought together in a hybrid, they segregate or separate during formation of gametes. Now segregate means what? It is separate. Okay, when a pair of of character, these are the pair of characters, is brought together in a hybrid. Now this one is the hybrid. Okay, which one? F1 is the hybrid. When it has brought in an hybrid, their gametes will separate. Okay, when how their gametes are separate, capital T is different, small t is different. It doesn't mean both are together in one parent. It doesn't mean that both are getting transferred together. Are they getting transferred? No, both are getting transferred separately. If both were getting transferred together, we would have got some medium sized plants. Blending we would have found, but that blending we don't find here in this cross. Okay, blending was not found in Mendel. What do you mean by blending? That is mixing. Okay, when we have crossed tall with a dwarf, okay, if there was a blending, we would have got a medium plant, but we didn't find a medium plant. Mendel didn't found there was no blending. Why it was not blending? Because there was a separation of the gametes. But when we have got a heterozygous tall, something like this, heterozygous tall, it was having both dominant and a recessive, tall and a dwarf. But which one was expressing? Dominant was expressing. So when it produced a gamete, two different types of gametes were produced. Both were reacting, both were getting crossed separately, not getting mixed. Okay, so that they are not getting some medium varieties. So to explain that, here he has given law of segregation. That law of segregation is this one. When a pair of character is brought together in a hybrid, they segregate or separate during formation of gametes. Example what? Again same cross what we have discussed just now. While explaining what you will do, you will explain about the separation of the gametes. During the separation and formation of the gametes, during that what you are going to explain, that depends on law of segregation. Now one schematic representation, okay, it is useful for three definitions: inheritance of one gene or monohybrid cross, law of dominance, law of segregation. Okay, all the three are very important. 5-mark question, any one of that will be asked. Okay, explain monohybrid cross, explain law of dominance, define law of dominance, define law of segregation, define law of segregation with an example. What will be an example? Same monohybrid cross will be an example. A complete cross till F2 generation till phenotypic and genotypic ratio. If you don't write phenotypic and genotypic ratio, you'll lose marks. 1 or 2 marks will be lost. Okay, so very important it is. And the definitions which I gave, it's not necessary you must write that only in your textbook on page number 75. Okay, you have here principles or laws of inheritance. First law, law of dominance, and the second law is the law of segregation. These are the true laws. Law of dominance, 3 points they have given, that you can write, and law of segregation, what they have given, you can write that too. Okay, both are there, you just go through. Okay, definitions what I gave, you can write that if you want. And the cross is similar. If you write this cross and practice this cross, it will be very easy rather than going through the textbook and getting confused. Okay, then I have used the term homozygous and heterozygous. Homozygous is individual having similar genes in an allelic pair. Example, what is that similar genes? Tall. Tall is having similar genes, what? Capital T, capital T. We have used dwarf, it is having small t, small t. For this we call it as homozygous. The heterozygous is that individual having dissimilar genes in an allelic pair. Example, tall character. Tall, which one? Capital T, small t. These are having dissimilar genes in an allelic pair. These are having similar genes in an allelic pair. If dissimilar genes in an allelic pair, heterozygous. Similar genes in an allelic pair, it is homozygous. Okay, these definitions also has been as differentiate between homozygous and heterozygous condition. Okay, so very important today. Today's class, what we have discussed: monohybrid cross, inheritance of one gene, law of dominance, law of segregation. Okay, go through your textbook, go through my notes, so that it can very easy to it can be very helpful for you all to understand. And factors will be used while studying something related to Mendel. So do remember, wherever you get factors, that is nothing but genes, which we know today as a genes, but at that time during 19th century Mendel was not knowing about genes, which means genes were not discovered yet. Okay, what are genes? Genes are nothing but units of inheritance. Okay, about genes and all I shall discuss more in my next chapter further as we are proceeding with the other topics. Anyhow, this is about today's session. If you have any doubts, any queries, please comment. Very important session it was, and very easy it is. Don't neglect it. And in my next session I am going to discuss about the test cross. That is very short, so I shall start with that. And if you have if you want any something other, I shall start with that too. And stay tuned till my next video. Unique bio classes. Stay safe, stay healthy.